Assalamualaikum warahmatullahi wabarakatuh. Good afternoon. We are from Group 4. Today we're going to talk about leaf blight in rice plant. Before we start, let me introduce our member. First is Sara Vanesya A, student number 131. Titin Andriyanti, student number 135. Dewi Istikomah, student number 137. Dena Aisyah, student number 138. And me, Rinta Sofia, student number 145. First is bacterial leaf blight. Bacterial leaf blight, or HDB, spreads in various ecosystems of rice plants and is difficult to control. This disease is caused by the gram-negative bacteria Xanthomonas oryzae PV oryzae, which produce blight that damage the chlorophyll of leaf and causes a decrease in ability of plant to carry out photosynthesis. Symptoms and the effect of leaf blight in rice. When the attack occurs on the beginning of growth, the plant withers and dies, and this symptom is called cracker. If this attack occurs in the flowering phase, it will interfere with the grain filling process, causing the grain not to be filled completely or even empty, so that these conditions cause a very large yield mortality. Bacteria that cause leaf blight in rice. Bacteria that cause leaf blight in rice is the bacteria Xanthomonas oryzae PV oryzae, or also called zoo. Zoo bacteria have quite a lot of pathotypes. The dominance of pathotype between different regions can be different. The distribution of pathotypes between different regions can be different.
Is the effect of the XOO bacteria in rice varieties. Pathotype changes can cause a rice variety that can initially resistant to HDB to become susceptible or fracture of resistance. This is influenced by several factors such as the composition and dominance of the pathotypes, and the rate of change in the pathotypes, the frequency of planting, and composition of differences in genetic background of the varieties grown in a certain time and range. Next. How to control leaf blight in rice? Part 1 is one way to control leaf blight in rice is to regulate the planting time of rice in irrigated land based on the time of distribution of water. The planting schedule varies depending on the position of the rice field relative to the main channel, namely the upstream or head, middle, and downstream. Next. How to control leaf blight in rice part 2 is on a cropping schedule perfectly suited to disease progression. The beds will become epidemic for certain diseases. Therefore, information on the composition of the pathotype and the dominance of a disease in a rice ecosystem, spatial and temporal, is important as a basis determining disease control strategies. Next. Sick leaf sampling collection. HDB infected leaf samples were taken at rice plantation in areas with 1, 2, 3, and 5 irrigation times. The location chosen is the area with the position of the rice planting toward the main canal, namely in the upstream, middle, and downstream areas.
Next. Location of rice planting with irrigation time. Paddy field with irrigation time 1 is the location of rice cultivation, which is upstream. Paddy field with irrigation time 2 and 3 are rice planting locations that are in the middle of the water distribution. Paddy field with water distribution time 4 is the location of rice planting, which is downstream or at the and of the distribution of water.
Next, sampling method. Sampling method of Howard down leaf infected leaves using stratified sampling with for its different irrigation system represented by at least 3 places. Each street is divided into 3 sections, each part of which is used as a repeat. Its replication was represented by 5 sampling points taken diagonally, where at its point one leaf symptomatic of HDB was taken. Next. Put 6 leaf sample into a paper envelope and then record information about the sampling location, rice varieties, plant stadia, and disease severity using the scoring method. Score 1, attack 1 until 6% like yellow left only an indication of disease plant has no symptom. Score 3, attack severity 6 until 12%. This is the leaf has necrosis milk. Score 5, attack severity 12 until 25%. This is moderate. Score 7, attack severity 25 until 50. This is severe. And score 9, attack severity 50 until 100. This is very severe. Furthermore, the leaf sample with HDB symptom were taken to the laboratory to isolate the zoobacteria. Next.
Cobacteria isolation. Isolation of the cobacteria was carried out in the laboratory using the leaf washing method. Cobacteria isolation step. First, cut a sample of sick leaf about 1 millimeter in size. Second, then wash with sterile distilled water. Third, collection water from washing leaf infected with HDP in a Erlenmeyer glass. 4. Dilute the using washing water to 10 to 6 power dilution, then take 1 cc of solution, then plant it in a petri dish containing medium potato sucrose agar or PSA. Next, cobacteria incubation stage. Incubation of the sample was carried out in the laboratory at room temperature, and observations were made every day to determine the emergence of cobacteria colony. Subsequently, the single colony of that emerged were transferred to standard PSA medium and then multiplied for the purpose of identifying the pathotype. Next.
And the next, inoculation and disease observation stage. Inoculating XOO isolate tested on differential varieties by means of the seizure method at the time of planting before the primordial phase. Step of inoculating bacteria. 1. Cut the tip of the rice leaf along 10 cm later. 2. Keep the leaves in a bacterial suspension for 48 hours with a concentration of 100 XCVU. Inoculation is carried out in the afternoon so that the result plant does not get too high a temperature stress. Next. Observation of disease severity. Observation of disease severity was carried out by measuring the lack of symptoms at 15 and 30 days after inoculation. Next. And the last, next ring. And the last, disease severity is the ratio between the lack of the symptom and the length of the leaves. Variety resistant reaction were grouped according to disease severity at the last observation. That's all for today's presentation. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.